we have a technology to make a couple thousand people's iPSC at the same time in the same room. So in that case, the fixed cost can divide it that numbers. For example, maintenance cost is, for example, $1 million. If we can make 1,000 people, we can dramatically reduce the cost to I think couple 20, 30 or 40K US dollars. So that's the first step to pricing down for the manufacturing.
Hi, I'm Jeremy, founder of the regenreport.com. We cover the regenerative medicine industry. You may have heard of induced pluripotent stem cells, or iPSCs as they're known. And if you haven't, I'll give you a really short version to start off. So, iPSCs, the discovery of them was actually such a big breakthrough that it went on to win the Nobel Prize years later. Essentially, what they do is they take adult cells from your skin or other cells, and they reprogram them in a lab to make them behave like embryonic stem cells. And these stem cells are really powerful. They're really versatile. They can change into basically any tissue in the body. So it's very powerful stem cells that they can make from your own cells. And today's interview is actually with one of the early pioneers of iPSCs, and that's Dr. Koji Tanabe, who is the second author on the human iPSC paper, and now he runs a company called iPiece. And in this interview, Dr. Tanabe is going to run through some of the potential applications of iPSCs from stem cell therapies to drug development to longevity, what led up to the discovery of iPSCs, how his company is bringing down the cost of manufacturing those, and how iPSCs may transform the world's healthcare. And if you like this sort of content, I do a weekly newsletter on regenerative medicine. I'll put a link down below. You just pop your email in here and we'll send that to you. And if you wouldn't mind liking and commenting and sharing this video so we can help push the regenerative medicine message out to more people. Thank you. Enjoy the interview. Can you describe the first time that you heard about stem cells? Where were you? What was going on? What piqued your interest?
So actually, so that's I think that's I think why I start this research. And I'm very interested in the newts or lizard rejuvenations. So because they cut their arms and they can rejuvenate, and fingers and eyes and heart, they can rejuvenate. And that time, because they can deprogram their skin or these somatic cells to the stem cells. So that's the first time when I heard the stem cells. So I'm really interested in about these regenerations. So I want to know, right, because how to regenerate these limbs or I think heart, and if because I was a high school student, so that's why I saw that I think newts or lizard can do that. Why not the humans? Why not the mouse? So that's my starting point. So that's my first time when I heard the stem cells.
That's interesting. I think that there are many people that had a similar start to that too.
Oh, really?
Yeah, I've heard from other scientists as well that yeah, that's interesting. And then you took that and you went on to work with Dr. Yamanaka. You started working in his lab. Was that in college or?
Yes, so that's why I think as I mentioned before, I really interested in these regeneration, the differentiation. So when I got the bachelor degrees, I tried to find some a place which we can research that is reprogramming. But at that time, no one have never ever done the reprogramming. I'm writing a letter and a couple, I think a lot of professors and that's 2024, 2004. And Shinya said, "Hey Koji," I think I meet with him the 4 times, and then he said, "Hey Koji, and if you come to my lab, and then probably we can work together." And that's the starting point for me. But at that time, I think nobody knows an iPS, nobody can emerging. We succeed to make IP, the reprogramming from the somatic cells.
Yeah, and then the next step, so leading up to that day, when you first realized like you had it, and maybe you looked through the microscope and saw the cells, the colony differentiated, or what had you, whatever gave you that signal that was like, we have this. Like, what was that moment like, and what was that day like? What was the mood like in the lab? Did you know it was going to be
Mm.
this big?
Uh, Actually, to be honest, that time, because that's a really secret, even in lab. Because when I go to the when I join the Shinya's lab, and I think I'm gonna answer that to your two questions at the same time. When I jump in the Shinya's lab, and the Shinya called me 2 days or a couple of days, I forgot, a couple of days after my joining. And Shinya said, I think, "Hey Koji, this is really secret, and only I think 2 people or 3 people know, even in the lab." And that's a world first mouse paper when I lead. I think, but I got when I lead that paper, that's a mouse case. I I'm feel my feeling too. I think one is I really surprised. I think, and then second is I think I'm really uh, uh, disappointed because I tried to do that job in my life, but he have already done in a mouse. That's my starting point. And then she has said, "Hey Koji, so why don't you I think work with the Kazu? Kazu is the first also in both mouse and humans paper, well, the first mouse and humans iPSC paper. And Koji, why don't you work with him to establish the human iPSC? That's my starting point. And my Kazu, but the first I think 3 to 4 months. It was really difficult for us to make iPSC. We use the same reprogramming factors when we reprogram the mouse, but that we couldn't succeed. We tried, I think, 100 times, we couldn't succeed. But I think my contribution as a second author is I think I find the way how to introduce these reprogramming factors into the somatic cells very efficiently. And Kazu tried to find, I think, another factors. But I think approach is try to increase the induction, the methodologies to introduce these deprogramming factors. And then once I think we have done these deprogramming and we got iPSCs, human iPSCs, we are really surprised. I think so because then I think because surprising, we saw I think we need a more factor to reprogram the human, but I think that we succeed to make a same four factor to the human iPSCs. But still, I think we couldn't believe that. And that's why I think we are really carefully test whether they have a functionality or they can differentiate into the 3 germ layers or that's, I think, markers, everything. So that's why that's gradually, I think we make a confidence. We have already established human iPSCs. But, that time, that's competition. And then two couple teams, Harvard, Wisconsin, and Kyoto University is a really hard, I think, competition at that time. And we immediately start to, I think, like the paper. But we published the paper, the Cell paper. But the Wisconsin team, Jamie Thompson, published a paper to the Science paper. And they tried to match up in the same day to, I think, publish the paper only that week.
Oh, wow.
That's
Okay.
it.
You had a race.
Yes.
Okay.
So that's like not amazing. We are just busy to publish the paper as soon as possible. Yeah.
That's awesome. And then as iPSCs, the Nobel Prize is awarded, and then everyone learns about them. And as you can see, there's a lot of potential. Um, Immediately, what were some of those first challenges that you saw, whether it's cost, manufacturing, stability, that sort of thing? What was the first thing that you're like, we need to work on this?
So the 10, 20 years ago, or at this time, the 20 years ago, right?
Oh yeah, but basically right after it was after the breakthrough.
Yeah, so the first challenging is in the efficiency was really low, right? Because the first reprogramming efficiency less than 1%. We see that we can reprogram that less than 10 colonies from the 1 million somatic cells. So that's I think starting point, and that's the first efficiency. And the second is safety. We are using integration methodology to make iPSCs. That also make damage to the DNA. They trigger some tumorigenesis after transplantation. The second. And the third one is, I think, making iPSC as a clinical grade. That's a third step. But first, I think a two-step is, I think, a scientific improvement is essential. So, but that this 20 years, I think people, I think scientists is overcome these, I think, these point because we nowadays, if we use, even in our company, if we use our technologies, I think majority of the somatic cell can become iPSCs. The program efficiency is almost 100%, near to 100%. And also, the integration part in we are using recently not only us and but also I think all the globally people use the integration free methodologies to make iPSCs and also clinical grade. And we are people start to already understand how we should establish the quality, how we should confirm the quality of iPSC for transplantation. So that's I think a little bit first challenging as I think a science. And then as you mentioned, next is I think a cost and also mass manufacturing. So that's why I think I found the companies because I like iPSCs, I love iPSCs, but I think I want to bring down this technology to the bedside as soon as possible because I think I don't want to keep saying iPSC is promising technology only to get a grant. Of course, that's a really great excuse to get a grant from the government. But only I think I don't want to use and really want to help the people by leveraging this technology. That's why I think I found the company and to tackle the issues of the mass manufacturing with the low cost. So that's a second the problem. Second issue, second I think obstacles that we almost try to overcome the obstacles. Yeah.
Awesome. Yeah, I mean, the cost is directly tied to accessibility, and that's directly tied
Yes.
to how it gets rolled out and how you help people who need it. So, um, could you explain a little bit about the economics of making iPSCs and how much it costs and what you guys do to sort of mitigate that, and we can sort of introduce eyepiece in there as well.
Yeah, so that's a really depends on the qualities. But I think well, let me I think that dive in let me approach to that questions from the manufacturing and mass manufacturing side. So because so far it was really difficult to it was because people I think making a take a people took a uh, spend a time almost 1 year or 6 to 12 months to manufacture the iPSC from the single donors because the reprogramming and QC and then making a master cell log and a QC and this is going to take a long time. That's why it was really difficult to mass manufacturing. But if we make some semiconductor manufacturing, we can make a lot of semiconductor at the same time in the same room. But on the other hand, these cell manufacturing, we cannot do the multiple manufacturing in a side by side in the same room because people concerning about the cross contamination each other. So that's why I think we should do one by one. That's why I think the 1 year per one person is I think historically 1 year one person. So, but I think our innovations, I think we are making, we are, I think, we have minimized these clean room into the small box. And if the since this small box is a cross clean space, we don't worry about any cross contaminations each other. So we can manufacture the couple hundred, couple people's I think iPSC in the same room at the same time. So we are succeeded to manufacture the couple. I think we at least we have a technology to make a couple thousand people's iPSC at the same time in the same room. So in that case, the fixed cost can divide it that numbers. For example, maintenance cost is, for example, $1 million. If you can make 1,000 people, we can dramatically reduce the cost to I think couple several 20, 30 or 40K US dollars. So that's the first step to pricing down for the manufacturing.
Awesome. Yeah. So yeah. I mean, if you talk about semiconductors or electronics or anything, it's like you build that prototype stage, and building that prototype car or whatever is really expensive. But not until you get to that economy at scale,
Yes.
where you can manufacture many of them, can you actually bring it down
Yes.
to a cost that makes sense. So that's
Yes.
generally what you guys are doing.
Yes, it's true. So because as you mentioned, because previously, for example, car too, right? Because of before the Ford, I mean, people make a car one by one. It's a handmaking. It's really expensive. But I think Ford succeeded to make a car like I think as a line, and then people make parts, and then I think people put the engines, and if they make this process. And at the same time, the people make a process to manufacture a couple, I think the couple, I think these product at the same time in the lines, people can reduce the cost. That's the thing we are doing the innovations. And also, as you mentioned, I think iPSC is similar to the semiconductors because iPSC itself is an insignificant cell product. People doesn't want to transplant iPSC itself because it make a teratoma. Because if we make neurons or cardiomyocyte or cartridge from the iPSCs, that's I think people want to get. But I think if the iPSC is essential as a starting materials, that's why we are always saying, right? That Intel always say Intel inside, but iPS inside your body. So because iPS derived cell product become I think that's I think iPSC can become neurons or cardiomyocyte or the skins or everything, the iPSCs, derived cell product can contribute to maintain the people's health.
Awesome. Um, and can you also describe, so you're doing that and you're making personalized iPS cell lines for people, like banking sort of
Yes.
lines. Could you describe that?
Yes, so by leveraging our mass production capabilities, at this time moment, we start the personalized iPSC banking service around, I think, depends on the service, but around the 40K US dollars in the US. We are, and that's, I think, probably we can, I think, so far in the U.S. market, people is banking the cold blood when the kids when they got the babies, but the cold blood can only think cure the leukemias, but mainly. But on the other hand, if they can save the iPSCs, they can I think people can approach to the couple of disease, and also. And also one more thing is the cold blood. Of course, even if I want to get my own cold blood, it's really difficult. I don't have an umbilical cord. But iPSC technologies, we can make these type of the age-reverted stem cells from anyone. And also, I think we can reprogram that 80 years age to the fertilized egg state. And people, anytime people can access their or starting, starting a cell state.
Fascinating. And then as as stem cell transplantation research gets better and better, if let's say hypothetically we find that, mesenchymal stem cells through IV injection is perfect for this sort of indication, we could always take those iPSCs out of your personal cell bank, differentiate into an MSC and use that for that.
Yeah, so we are, and also that's a really great point. And we are trying to also, I think, approach because so far iPSC is iPSC application. People say regenerative medicine or I think drug discoveries, personalized drug discovery. But as you mentioned, that iPSC can be leveraged for the longevity and rejuvenation too. So as you mentioned, because iPSCs, I think, have a three features. One is I think we can expand as many as we want. We can get any type of somatic cell. Third is we can get your age-reverted stem cells. The MSC's case, you can, if that person is 60 years old, people get the 60 years old stem cells. But iPSC's case, you can get the 0 years old, I think, stem cells. That's a completely different. I don't know whether the iPSC is going to replace or not, but iPSC is can make another effect from these MSCs, I'm sure.
Interesting. Yeah, it'll be an interesting market to watch in the next 5, 10 years once more research comes out because you got a lot of people that fly to Mexico to get like Wharton's jelly or other young allogeneic sort of cells because of what you just mentioned. My cells are 32 years old, and you can get younger ones, but with the allogeneic, they don't have the HLA like class 2 markers. So like maybe it's not rejected, but I don't know if they actually know that for sure. But if you were to differentiate your own iPSCs,
Yes.
maybe that would even be better.
Yeah.
Any
For example, like a people called I think rejuvenated T cells, T cell NK cells, because so far people in a market people expand their own primary NK cells in outside of the bodies, but during the expansion they exhaust it, and since that's I think 30 years old or 60 years old NK is NK, but that these NK cannot attack a cancer. That's why they're suffering from the cancer, right? So that's why I think iPSC approach, they deprogram and rejuvenate their blood cell to the iPSC and then re-differentiate into the immune cells, which can attack the cancer. Then people call the rejuvenated immune cells. The age-divided stem cell derived immune cells can attack the cancer much, much higher than the original immune cells. That's also we can, of course, I think you leverage that technology to cure the cancer. And also, I think we can leverage that technology for the longevities too, because the aging, one of the aging is getting back the immune system. But if we inject the age-reverted these immune cells, they can maintain the, I think, healthy immune system in the body. So not only the immune system, but also for everything, neurons and cardiomyocyte and the other stuff too. I think, yeah.
great. And then you touched a little bit about using iPSCs for drug development and research and such. So what will you potentially use them for, like organoids, or how would you leverage those iPSCs for that through your company?
So historically, as you know, because if we historically a drug discovery is during the drug discovery, people should test that drug in the some cells, right? If people I think test a mouse, but the mouse is different from the human. We don't want to get the drug which can cure mouse, and that's the first problem. And the second, even if we got some patient-derived primary cells, that's limited, and also that drug is effective to that person. We don't know whether that drug to effect to everyone or to you. So that, but on the iPSC's case, we can reprogram your blood cell to the iPSC and then differentiate in the targeted cell, like neurons, for example, neurons or cardiomyocyte. For example, if you're suffering from the Alzheimer disease, get the blood and then make iPSC and then iPSC and then make neurons. We can directly test the drug using your neurons. because we can't. That person is surviving. Hey, I think please give me your neurons from your brain. People say no, I don't want to get a cut in the brain. And that's a no. But iPSC technology can, I think, can enable to get the access to the patient, the person. The person's own, I think, the somatic cells, any type. So that's why I think people try to find a drug. And in Japan is already they making iPSCs from the ALS patient, and they differentiate the neurons, and then they find some new drugs, and that's already in a drug, I think, a clinical trials phase 2, and it showed, I think, very great efficacy to stop the progression of the HLA. Like that, I think we can use the iPSC for the drug discoveries.
Wow.
But not only a drug discovery, from my understanding, for example, like a cosmetics too. If we make iPSC derived skin or organoid or 3D, I think, skins, we can test which cosmetics is suitable for that person, or I think which cosmetics is not make a toxicity or allergy to that person. So that's, I think, we a lot of people is focusing on only the, I think, the medications, but of course we can leverage these iPSC for another longevity and beauty application too.
Awesome. Are there any other applications for iPSCs that are not really talked about? Anything else that maybe people don't know about?
So that's, I think, we try to approach, I think, to the longevity at this time moment. Really, I think we try to approach because uh, again, I think we can get the access. Let me start these stories because couple, I think, 20 years ago, around 20 years ago, scientists connected two mice. The two mice is, I think, a circulation system. They connect the blood vessel in the two mice, and then one is young mice and one is old mice. They connected old mice getting young and the young mice getting old. Like that's a, that from that people learn that some young mouse have some rejuvenation factors, I think, factors in the blood or in the bodies. So that's why I think people, I think, try to find it, but people cannot get their own HB stem cells. I cannot get my own, I think, my own fertilized egg or my own, I think, young age cells, right? But I think, but on the other hand, if we make iPSC, we can get the access to age-reverted cells. So that's why we are focusing at that point. That we try to identify that these rejuvenation factor from the iPS. That we almost identify that. And in Japan, we started this service using we call it iPS factors, which is extract from the iPSCs. We can see a lot of great, I think, rejuvenation efficacies from these iPS factors.
Awesome. And you've touched on Japan a few times. Obviously, that's the home of iPSCs and a lot of regenerative medicine as a whole. And I've seen some news, occasionally you see news coming out of Japan where it's some of these iPSCs are already being deployed and maybe even covered under national health insurance for, I think, retinal, some
Yes.
sort of retinal condition. So could you talk about like what sort of
Yes. So let me pull up the couple applications slide. Hmm. As you mentioned before, in Japan, one of the remarkable recent achieve is this Dr. Sauer's group in the Osaka University is transplant the cardiomyocyte sheet onto the heart failure patient, and then they have already done the clinical trials, and then for the couple patient, that they can see the efficacy and the safety, and then one of the couple people start to work again. So that's why the Japanese government is almost approved to get these to for this cell therapy. That's the first one. And the second one, as you mentioned, another Osaka University succeeded to make these corneal cells derived from the iPSCs, and they transplant again the 7 or 8 people. The majority of them get the recover from zero point. I don't know that almost cannot see that their granddaughter grandson, but they can see after transplantation. They cannot see any I think concerning about the safety, and after 2 years, I think they follow up. They have because that's I think but couple years ago, people think iPSC may be significant to cure the disease, but that's already proved. The these cardiomyocyte and corneal cells. But not only that, but recent this year, Keio Universities, Hiroyuki Okano is transplant the spinal transplant the iPS-derived NSC to the spinal cord injuries patient. That's, I think, amazing. They already explained that data a little bit this year's the Japanese Society and also ISSL too, and they see the very great efficacy. So I think I got a surprise. I don't know how much I can say, but that's a game changing because that so far they cannot move, but after transplanting iPS-derived NSCs, it's changing.
Yeah, I saw that. That was just a couple months ago. Apparently, he like stood up out of his wheelchair. That's crazy.
Yeah. Yes. So that's crazy. That's also my motivations, right? Because because as you mentioned, MSC or allogenic, of course, I think they are the two type of the regenerative medicine. One type regenerative medicine is, I think, just transplant something, they secrete, but even that cells is rejected by the immune system, but that cells is just work as secreting some growth factor or something, some cure that one. But another real rejuvenating medicine is contribute to recover the functionalities. In that case, that these cells should be survive for a long time. So in that case, that should be the autologous is the best. And allogenic is maybe okay in some specific organs or tissues. But I think we want to, I think, contribute to these and real, I think, of regenerative medicine to recover the functionalities. So that's why I think that's these NSCs transplantation is also, I think, encourage us to contribute this to create the world which people normally have their own iPSCs.
Fascinating. Yeah. There's so many things that we have no answer for at all. Traumatic brain injury, spinal cord injury, many other ALS, Parkinson's. I mean, you name it. There's a lot of things that there's
Yeah, the US too, right? Because US also, Blue Rock also, I think, published a paper who succeeded to have done the clinical trials. That's the ESCs, but I think they have already done to do the clinical trials for the Parkinson's. And also Vertex also have done too. That's also really, I'm very impressed. Vertex also succeeded to transplant iPS-derived beta cells to the diabetes patient, and they can see the efficacy and the safety. That's also because they should inject the insulins, but they can at least decrease the number of the insulin injection. That's before you can increase the quality of life. And yeah, so that's I think thanks. But this orange is I think everything done the clinical trials in the globally. So that's why people are thinking iPSC is still research stage, but this orange is already started or have done the clinical trials in the world.
wow, that's interesting. Um, and kind of back on that, how do you see the regulatory landscape evolving in Japan right now? I've also heard some reports from some people saying there's been some talk about pulling regenerative medicine back, and then other times seems like it's going full steam ahead. Here in America, it's hard to tell. Is there any other insider insights that you could give?
Yeah, so the even in Japan, the iPSC derived cell product is really restricted. So of course we should I think go through a lot of discussion or I think approval from the regulatory agency, same as US. The US side too. The US is also is we should going through because that's a little more difficult than the primary cell line MSCs or these guys because MSC or PRP's cases that's a minimum manipulations, but iPSCs is we are introducing factors and we are, I think, reprogramming the sulfate, and we should be more careful. I understand. So that's, I think, that's ongoing, and both in Japan and US, and also that regulation is makes sense for me because the reprogramming is making a lot of, I think, a stress to the cell product. We should be careful whether we don't have any mutation in the genome or we don't have any some abnormality in the cell features. We should be, I think, a test. And that's why I think in our I think a product, of course, we are doing the cancer target sequence, cancer panel sequence, and also to understand that these genomic stabilities, and we are really carefully to check that these quality too. And also during in the manufacturing, of course, we are following a 21 CFR 210 to 11 ICHQ7. That's FDA's guidelines. And USERX and Europe side USERX 4. That's really important to make sure to help the people. So because if we inject something wrong, I think a cell product to the patient and then integrate it in the patient's, I think, bodies, it's ridiculous. We should be, I think, be careful, and we should be, I think, make, I think, much higher grade of the iPSC product.
That's probably difficult to balance because on one hand, it has so much potential to help so many things that for people that are
True.
suffering terribly that have no answer at all. But with any new technology, and especially, I mean, any new medicine, something bad eventually is going to happen. Medicine's never perfect, but with anything brand new like this, something small can happen, and it really scares people much more. It can kind of blow it out of proportion. So what what else do you guys do? Is there any sort of testing that you do post manufacturing to ensure the stability and quality of the cells? Can you give a little intro to that?
Yeah, so again, so we and also FDA and also people in the field, I think people start to understand, people start to get the experiences about the balance, right? Because first, iPSC will not inject into the body. iPSC is one of the starting material, raw material of the final product. The final product is a neurons. Final product is iPSC, the neurons, or I think muscle cells, or cartridge, or other cell. That's it. We should, I think, we'll make a deep QC in this final product. And then iPSC's case, and we can, I think, make a balance as a starting material. But still, iPSC is essential, I think, starting materials, and they are a key component of the final product. But we don't have to make a redundancy in the all the time. So that's, I think, otherwise, because if that's not much to the marketplace, the iPSC cannot, I think, as you mentioned, iPSC cannot, I think, coming down to the people's hand. So that's a really important balance. And also if we that's why I think process is really important. And then we once we validate the process, and we have a if we have a process which we will not make which will not make a genomic stability, or I think a bad grade of the iPSC, we don't have to make this deep QC because we the process have already validated. And that's also another important point to reduce the cost. Not only we should not validate the final product, we should validate the process too. Yeah.
Great. Um, and then for timeline's sake, so I mean, it's probably hard to say right now with regulations and technology, but generally, how long until you think stem cells derived from your iPSCs manufactured from eyepiece are at the bedside or being deployed to patients in the US?
In the US, I think we try our best within the 3 years that we try is doing, but we have already transplant the iPS-derived NK cells into the outside of the US. That's, I think, one of the first clinical trials in using our iPSCs. We transplant these iPS-derived NK to the ovarian cancer patient. Before injecting these, I think the NK cells, that these patients have a huge abnormal waters, but that's it. After injecting iPS-derived NK, that's a suppressed, clearly suppressed, and also clearly stop the cancer progressions. And I think that's, I think, already done in the outside of the US. We are now next step. We are, I think, leverage these data to start clinical trials in the US as soon as possible.
Awesome. What do you anticipate? You think that you'll also be doing NK trials, or you do other things for this first few trials, or anything else you think it's going to be the first things that on the horizon?
Not only the NK, sure, but I think, of course, and also I think, yeah, we cannot do everything by ourselves. And that's why we are providing the iPSC to the partners, and the partner also will, I think, as progressing the clinical trials. I think using our iPSCs, we already provide our iPSC over the 30, I think, partners, and who is, I think, focusing on the clinical trials. And then they are ongoing to go to the clinic. And at the same time, of course, we are going the clinic by ourselves too, by leveraging our iPSC itself. But this is only regenerative medicine. But I think, of course, we want to, I think, progress on rejuvenation too, using iPSCs. So that's the next step.
Excellent. Um, those are pretty much all the questions about eyepiece and iPSCs that I had for you. Is there anything else you wanted to talk about the company or about the technology that we missed?
Uh, I I talk about a lot. I talk a lot, but basically, what we want to do is right? Because so as I mentioned, because what we want to, I think, because yeah. So let me put up another slide. I know, I think no, it's right here. Yes. But anyway, so because the medication is starting from the Chinese herbal medicine, right? And people using these hard leaves and roots, or I think that these things that people use these plants as a medication. And second, people start to use a small chemical compound, antibodies, and RNA as a medications. But the next step, people start to use a cell product as a medications. And for example, if they are suffering from the cancer, and of course they are, I think so far people is using chemotherapies to cure the cancer. But recently, nowadays, people study the T cells or that these immune cells to attack the cancer. But the most important things, I think, nobody, I think, stepped on a challenge for the mass manufacturing. People don't know how to mass manufacture these cell product. That's why we try to tackle that problem to mass manufacture these cell product. And then that's the first step. I think we are tackling that with the iPSC, but we are not only focusing on iPSCs as well. And and also I think we are again the iPSCs is platform technologies. We want to realize the future which people normally their own iPSC when they are born, and they stock the iPSCs, and then once they got some disease or I think injuries, and also I think if they want to go get the longevity service, then I think they can use their own iPSC for these applications. That's I think I want to realize that these futures for the people.
Awesome. Great. Well, yeah, I mean, those are all the questions I have for you. And I really appreciate your time. I've learned a lot. So Dr. Koji, Eyepiece Inc. And yeah, we'll call that the end of the interview.
Thank you very much.